Hello friends, in today's lecture we are going to learn about the different cell organelles present in the plant and animal cell. Nucleus, it is the main center of the center cell organelle of center organelle of the cell. It consisting of outer nuclear membrane and the liquid which is present inside the nuclear membrane is called as a nucleoplasm. Nucleoplasm is embedded with the nucleolus. Nucleolus performs it mainly helps in the rRNA synthesis, which is responsible for the protein synthesis. Where this true nucleus is present in the both the plant and animal cell, whereas in case of bacteria, which is a prokaryote, the true nuclear membrane and is absent, and you can see the genetic material which is compact in the form of, which is compact in the nucleoid region. uh, Here we can this ring like structures, these coiled structures are called as DNA, which is a genetic material in most of the living organisms. The genetic material can be DNA and RNA. DNA means deoxyribonucleic acid, and whereas RNA means ribonucleic acids. In the coming lectures, we are going to talk about the main differences between these two genetic materials and what are the types of DNA and different types of DNA and RNA and what is its main role. Uh, it mainly like genetic material is mainly confined for the transformation of the genetic information from one cell to the another cell. So the next important cell organelle is mitochondria. It is a double layer membrane consisting of the outer membrane and inner membrane, and the inner membrane is enfolded into structure into foldings called as cristae. And this cristae has this rachis particles or F0 or F1 particles, which is mainly responsible for the synthesis of uh, uh, which has ATPase activity. Particularly F1 is involved in the synthesis of mitochondria. This nucleus was first discovered, I forgot to mention, nucleus was first discovered by Robert Brown in the year 1833, whereas mitochondria was first discovered by Albert von Kolliker in the year 1857. The main function of the mitochondria is to carry out the oxidative reactions of the cell, uh, particularly electron transport chain, and it is a powerhouse of the cell. It means that it provides energy to the cell in the form of ATP, and the F1 particle is responsible for the synthesis of the ATP. This is a picture of endoplasmic reticulum. It was first discovered by Charles Garnier in the year 1897. It it cons it if the ribosomes can be sorry, the endoplasmic reticulum can be of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum means it consists of RNA containing granules that called as ribosomes. If the ribosomes are present, then it is called as a rough endoplasmic reticulum. If the ER lacks the if the endoplasmic reticulum lacks ribosomes, then it is called as a smooth endoplasmic reticulum. You can see this the presence of ribosomes makes the endoplasmic reticulum to appear very rough, and this mainly rough endoplasmic reticulum is mainly involved in the protein synthesis, whereas the smooth endoplasmic reticulum is involved in the lipid synthesis. Coming on to these two organelles, that is mitochondria and ERS, present in both the plant and animal cell, whereas uh, bacteria or bacteria lacks these organelles. The next important cell organelle which is specifically present in the plant cell is the chloroplast. Uh, it was first discovered by Hugo von Moll in the year 18. 37. So the main function of the chloroplast is to carry out the photosynthetic process. That is, it helps in the synthesis of food in the presence of light. It absorbs the light energy and it converts it into the form of chemical energy, and it synthesizes its own food. And the reaction that is mainly occurring in the uh, structures called as grana. These grana are the stacks of thylakoids. That is, these are the flats. In these individual structures are called as thylakoids. They are flattened sac-like structures. The, and the stack of thylakoids is called as the granum. The light reaction of the photosynthesis occurs in the grana, and whereas the dark reaction occurs in the stroma. It is the liquid portion is called as the liquid or the ground fluid, which is present in the chloroplast, is called as stroma. There are some Fred channels which are present in the chloroplast. As we know, this photosynthesis process occurs in the presence of carbon dioxide and light. Carbon dioxide is very important source for the photosynthesis process. So these Fred channels are helpful in the repair, carbon dioxide repair thing. That is, the Fred channels contain some enzymes which help to repair the carbon dioxide amount or content. So the next important organelle which is present specifically in the which is present in large number uh, in case of plants, that is a vacuole. The outermost layer is called as the tonoplast, and the liquid portion or the inner material of the vacuole is called as the cell sap. It contains enzymes, all the enzymes, salts, minerals, proteins. Some are present in case of plant are present in the plant vacuoles. The main function of the plant cell wall is to regulate acidity of the cell and turgor pressure of the cell. Acidity and turgor pressure of the cell and also transport of substances around the cell. Lysosomal and as well as this endocytic kind of the transport that occurs in case of vacuole. The next important cell organ is Golgi apparatus. It is present in both the plant and animal cell, whereas uh, bacteria, in case of bacteria, it is absent. Uh, it mainly helps in the protein folding as well as the protein packaging process. The synthesis of the synthetic phase is called as a cis phase, and the phase which is opposite to the cis phase is called as a trans phase. They are mainly responsible. This Golgi apparatus is responsible to secrete some kind of vesicles, wherein the proteins can be packed in the form of vesicles, and it helps in the and it helps in transport of those uh, embedded in the vesicles to different target sites. The space present in between the membranes there, that is called as a lumen. Cis phase is the receiving phase, whereas the trans phase is the exit phase. This trans phase is responsible for the transport of proteins out of the cell. So the main function is protein folding and protein packaging. The next important cell organelle which is specifically present in the animal cell, whereas it is absent in the plant, is called as a centriole. It is mainly responsible for the cell division, cytokinesis, that is division of the cytoplasm, and spindle apparatus formation that helps in the cell division process and cell separation process. The centrioles can be present in the form of microtubule triplets, that is nine the fibrils which are arranged in the outermost in the outer or peripheral layer, whereas the fibrils, if they are absent in the center region, then it is called as then it is represented as 0. So 9 plus 0 arrangement, or 9 plus 1 or 9 plus 2 arrangement kind of thing we can see in case of the microtubule triplet arrangement. Lysosomes are present lysosomes are present in both. Lysosomes are present in the animal cells. They're mainly consisting of the hydrolytic enzymes. They are also called as the suicide bags of the cell, as they contains digestive enzymes which digest uh, its it, it digests the content of its own cell. That is why it is called as the lysosomes or suicide bags of the cell. The next important, this is a ribosome. This subcellular component of the both the plant and animal cell includes ribosomes. Ribosomes can be of, in case of prokaryotes, they can be of 70S type, and whereas in case of eukaryotes they can be of 80S type. 70S type again is further divided into 40 and 50 and 30S type, whereas in the case of eukaryotic, ATS type is further divided into 60 and 40S type. Here the S represents the Swedberg unit, and it represents the speed of the sedimentation of the proteins in the centrifugal field. So the liposomes are the phospholipid bilayer, which are sometimes originated from the endoplasmic reticulum of the lysosomes. So, which are arise from the lysosomes. So this is the bilayer of the bilayer phospholipid is called as the liposomes, whereas a single layer of the phospholipids are called as micelles. These terms you have to just remember for the point of examinations, competitive examinations. The next important cell organelle is a peroxisome. It helps in the regulating the hydrogen peroxide content, and it is mainly responsible for the beta oxidation process. It is present in both peroxisomes helps in regulating the hydrogen peroxide content. It contains an enzyme called as catalase, which helps in the breakdown of the hydrogen peroxide. Both peroxisome and mitochondria are involved in ROS scavenging activity, as well as the reactive oxygen species production. So the difference between these two, like both the mitochondria and peroxisomes both carry beta oxidation process. The main difference between the main difference is that in case of mitochondria, flavin dehydrogenase does not react with the molecular oxygen, whereas peroxisomes react with the uh, molecular oxygen, and there is no catalase enzyme in case of the mitochondria to regulate hydrogen peroxide levels, whereas in case of peroxisomes it contains catalase enzymes that regulate beta oxidation process. uh, what is beta oxidation? Beta oxidation is a process wherein fatty acids are converted into acetyl CoA form. Okay. One more thing, the centrioles are helpful in the Centrioles are discovered by Boveri and Benetton in the year 1880, and these structures helpful in regulate in organizing microtubules, which are the important structures of the cell skeletal system. Okay, and Golgi apparatus was first discovered by Camillo Golgi in the year 18 1898. Plant vacuole was first discovered by Antony von Leeuwenhoek in the year 1676. So these are some of the important points regarding cell wall organelles of plant and animal cell. So in the next lecture we are going to study about the cell division process. Okay. If you have any doubts regarding this lecture, you can comment in the comment box section. If you like the video, please like and subscribe to my channel. Take care. Bye bye. See you in the next lecture.